So the next speaker is Russell Cox. Obviously, he's already on the stage, sitting actually, literally on the stage. Russell is from England. He decided to do an initial Brexit before everyone else from England, and coming to Germany is now in Hannover. And we're looking forward to your talk, Russell.
Thanks very much, Helga. It's a really great pleasure to be here today and to see a real audience. I feel a little bit like Elton John sitting here. Okay, I have my keyboard, but the reason I want to do it is because I want to use my cursor, and then the people who are online should be able to see what I'm pointing at. Okay, all right, let's make a start. So those of you who know me will know that I'm very interested in secondary metabolites from fungi. And one of the reasons for that is because fungi make chemically complicated compounds, and these often have, as we've seen in the previous talk, very interesting biological properties. So some of the compounds which we've been looking at in my group would include, for example, here squalestatin. This is a nanomolar inhibitor of squalene synthase, so a cholesterol inhibitor. Strobilurin A, this is a very potent antifungal, and it's used for the development of agricultural fungicides. Xanoviline is a potent agonist of the GABA receptor in brain tissue. And tenolin here, which doesn't really have any biological activity as far as we know, but we use it as a very interesting model. And of course, over the years, the kind of experiments we've done would be very traditional isotope labeling patterns. We can see where all the atoms come from. We can see the polyketides often buried in the structures of these compounds. And of course, more recently, we can sequence the genomes. We can do transcriptomics and get a lot of detail on the clusters themselves. But from my point of view as a chemist, those studies don't really tell us what we really want to know about the systems which make these compounds, and that is, how are they programmed, and could we change them? Could we engineer them rationally? And just like Eric, on the previous talk, I'm going to use the biosynthesis of erythromycin as a kind of reminder to everyone about how PKSs work. And everyone is going to be very familiar with this production line image that I'm showing here of the 6-DEB synthase. And of course, we know that each module adds one extender unit and will do the required amount of beta processing. And what I want to focus on here is module 4, because module 4 is kind of a full module. It has a full beta processing capacity. And of course, that's going to completely reduce this beta position here. So we're going to focus a little bit on module 4. Let's all remember that. And the reason also that module 4 is important in this talk is that module 4 is very, very similar to the vertebrate fatty acid synthase. And of course, there's a very nice crystal structure. It's been around for more than a decade now of the vertebrate fatty acid synthase. So we know a lot about the structure of the vertebrate fats. And it turns out that fungal polyketides take up the same model. Basically, highly reducing polyketide synthases in fungi look just like vertebrate fatty acid synthase. They look a bit like module 4 from the DEBS system. Okay, and of course we know a lot about the chemistry, how this works. Okay, there's an acyltransferase which is involved in loading acetate and malonate onto the system. Okay, the ketosynthase then of course builds this all-important carbon-carbon bond and makes the diketide intermediate, this beta keto compound. And then we get the cycle of beta processing. So for example, keto reduction, okay, to build in the alcohol. I'm telling you stuff you know already. Dehydration, enol reduction, and we make a fully saturated system. And then the key thing which is important here about the vertebrate fats, which is different from module 4 of DEBS, is that these are iterative systems. Okay, we have one set of proteins, okay, and the substrate is cycled around and around and around. Okay? All of these catalytic domains, as far as we know, are pretty dumb. They don't have any programming. They just take what they're given, they do the chemistry, and they pass it on. Okay? And so the system is not very intelligent, but it has to have some kind of intelligence. It has to control the chain length, for example, and that's done by the thioesterase. And the TE is able to detect the chain length, and it's able to hydrolyze the intermediate at the right length. So it's a pretty dumb system, and it makes fairly boring compounds. As a chemist, I'm not very interested in these. But we need to take this on board and bear this kind of chemistry in mind, because when we look at the gene clusters then of more complex fungal compounds, this is squalestatin, we can see genes which encode these types of highly reducing polyketide synthases. And if we look at the domain structures of those, they are identical to vertebrate fatty acid synthases. So we have the same domains in the same order. And if we take a gene like this, for example, and we express it in Aspergillus oryzae, then we can make relatively complex compounds like this tetraketide, in which there's clearly programming now in the system. Okay, we get 2 methylations, there's no methylation here. We get full reduction during the first 2 rounds of biosynthesis, but no reduction later. And of course, the chain length is controlled now to a much shorter length, although there's no thioesterase usually in these systems. So we have iterative systems which are, on the face of it, almost identical to the vertebrate fats, but which can make complex systems. There's one other difference, and that is in the polyketide synthases, we normally see that the methyltransferase domain is present and functional. So in the vertebrate fats, in mammals, in us, there is a methyltransferase domain in our fatty acid synthase, but it's broken. Okay? Much of the domain is missing, and it's clearly not functional. In fungi, that domain is present, and we believe that the methylation happens at this beta dicarbonyl stage, and a methyl group can be added. And then, of course, this beta dicarbonyl can go into the normal cycle of beta processing to make saturated intermediates like this. And of course, this is still iterative. Okay, this is one set of catalytic domains which is acting again and again. So there's different chemistry happening in each cycle. And if we look, for example, at the squalestatin hexaketide, then these types of synthases, although they look boring, they have to be able to select different starter units. They have to be able to program methylation position, and they have to be able to program the level of beta processing in each cycle. And those are the things that we want to investigate and we want to understand in my group. How do these iterative, very, very simple systems still make complicated compounds? How do they do it? Can we change it? I need to just introduce a bit more information. There are 2 main architectures that we see in fungi for these highly reducing systems. I've talked a little bit now about the squalestatin system. So here we see 2 polyketides made, a tetraketide and a hexaketide. They're both made by basically this type of polyketide synthase, which is almost identical to the vertebrate fats. We've got the same domains in the same order. If we look at other systems, like lovastatin, for example, again, there are 2 PKSs. One of them is identical to what I've just been speaking about, okay, and it makes this little diketide. But the other one is slightly different. Here, the enol reductase domain is broken, and there's a transacting enol reductase. So we see that quite often. Here in the case of strobilurin, this is another compound we've investigated in my group. We sometimes see some extra domains downstream of the ACP. This is actually very, very rare. Normally there's nothing downstream. But here, for example, there's a methyltransferase that we know introduces this methyl group here. In this particular case, the enol reductase is broken. Okay? And as you'd expect without an enol reductase, then there's a triene made as the intermediate. And this PKS does something that's also interesting. It can control the geometry, and that second alkene is put in Z. We don't understand how that happens. Okay, so there's one enol there's one dehydration here, and it can make E or Z under different circumstances. Systems like grafeldin, also very, very similar. Okay, this has actually got a trans-acting TE. And this TE is a little bit like the fatty acid synthase TE. It's able to detect chain length and control the chain length of grafeldin. And the final system I want to talk about today, which is going to come up later in the talk, are systems where we have a fused non-ribosomal peptide synthetase as the offloading module. And here we have one or the other of the 2 different types of fungal PKS. For example, in the case of fusarin here, the ER is broken, but there is no trans-acting ER. So in fusarin, we get this polyene type of structure. But yet again, this type of iterative PKS has to control the methylation pattern, and it has to control the chain length. So we've got a lot of programming which we need to understand. So my group take 2 main approaches to understanding these systems. The first one we call divide and conquer. And we basically want to clone and express individual catalytic domains, purify those proteins and study their enzymology using very traditional methods, okay, synthesizing substrates and figuring out which are substrates, which are not, what's the selectivity. On the other hand, we want to do a kind of engineering type of experiment, and we want to make chimeric systems. We want to take iterative TKS system we understand, or at least we know what the products are, and we want to make hybrid systems in which we're mixing and matching those domains and figure out whether we can change the actual chemical program of the system to make new compounds, and then work backwards to figure out what we must be doing. So I'm going to give you just a couple of examples of both of these types of experiments that we've been doing over the years, and some conclusions about how we think these systems are programmed and what is the mechanism. Of course, we're doing this against quite a difficult background. There is no structural data for any fungal iron-reducing PKSs. So we're using structural data from the vertebrate fatty acid synthase. In the case of the divide and conquer strategy, often these isolated domains are very unstable. You could imagine if we're expressing just a single domain here, we're exposing a lot of hydrophobic surface, and those enzymes then become pretty unstable and insoluble. So we can't examine every single catalytic domain that we want to. And we're going to use some modeling and docking really to make up for some of those deficiencies. I'll talk about that as we go. All right, so let's just focus on the first area, the divide and conquer. So we're going to work with the squalestatin tetraketide synthase that I introduced at the beginning. This is a really nice model system because all of the catalytic domains are functional at different stages in the cycle. Okay, during the first cycle, clearly we're going to make a diketide. It must get methylated. There must be ketoreduction, dehydration, enol reduction. So everything works in the first cycle. In the second cycle, everything works, methylation and full reduction. But in the third cycle, there's no methylation and there's only partial reduction. And for example, what we've done here is express the DH domain. So the DH makes up a nice dimer in the center of the PKS. We can express that, we can get soluble protein, we can synthesize all sorts of different possible substrates. Okay, we can control the stereochemistry, of course, we can control the chain length, and we can then test those. And here the assay that we use is an LCMS-based assay. We're just looking at the development of product over time, and we can plot that. And we get pretty simple data. So for example, looking at the stereochemistry, we can show that only the 2R3R intermediate is turned over, none of the others that we make get turned over. So these are highly stereoselective, but when we make longer intermediates, they all get turned over, as long as they have the correct stereochemistry. So we have to balance out the stereoselectivity and the substrate selectivity. We can build models. This is a very, very simple model of the DH, just built with a Swiss model, okay, nothing sophisticated. We can dock those substrates back in. Again, not sophisticated, just using auto-dock. And we get results where chemical moieties, which we know are involved in the mechanism, line up very nicely with residues in the active site, which we also know are involved in the mechanism. So the modeling kind of works, and it can explain what we observe in many cases. This is an example, a bit more sophisticated example with the enol reductase from the SQTKS system. So the enol reductase would be here, this green catalytic domain on the top. Okay, and what we see here is we're challenging this with a whole series of different synthetic alpha, beta, unsaturated thiol esters. And pretty well all of them turn over to some extent. So this domain can pretty well take what it's given, but some of those substrates can be processed very much more quickly. If we look at Kcat over Km, for example, the red bars, some of those substrates are processed hundreds of times quicker than others. We see, for example, that the tetraketide, the final product of this PKS, actually this is not turned over at all. Can't see any turnover here. And in fact, this compound turns out to be an inhibitor. It goes in the active site and it sits there and it can't be turned over. Okay. So we can get a lot of detail about how these systems work. So we understand then that domains have what we call intrinsic selectivity. They have their own selectivity. which they can apply during the program. Okay, this is obviously chemical selectivity because, for example, a dehydratase has to have a beta hydroxy system in order to work. Okay, but there's also pasted onto that then stereoselectivity as well. If we think about stereoselectivity, actually we have measured the stereoselectivity for every single catalytic domain now in this system, and we can show that every single stereochemical step is identical to the vertebrate fats, okay, both in terms of the cofactor and the substrate. And so really we understand these fungal systems as to be almost identical to vertebrate fats, except that they're programmed. Okay? Or you could think of the vertebrate fats as an unprogrammed polyketide synthase. They're the same thing. If we have this level of detail, can we reprogram these systems? And this is again just an example from the ER, the enol reductase. And again, this is a very simple model, just made using Swiss model and the SQTKS sequence threaded onto the vertebrate FAS sequence. We can then dock substrates into the active site. I'm sorry, it's probably a little bit small to see here, but what we observe in these models, for example, is that substrates which we know are good substrates, substrates which are turned over by this enzyme, take up an S-cis conformation, and molecules which we know are not substrates, in fact these are inhibitors, take up an S-trans conformation in our docking experiments. And it looks like maybe that's involved in controlling whether these can be substrates or not. We can also look at the pocket in which the polyketide is residing, and we can of course then see interacting amino acids. And in vitro, sorry, not in vitro, in silico, we can then mutate those and we can redock back into that pocket. This is very, very simple, just online docking using autodock. But if we, for example, make some mutations here in these phenylalanines, which are contacting the tail of the polyketide in this isoleucine, we can actually convert what was an inhibitor into something which should now be a substrate. And you can see the back in blue. This is the docked tetraketide in the wild-type system, and it's taking up this S-trans conformation. When we make the mutant in silico and we redock the same compound, then we very nicely get this S-cis conformation. And so we would predict from this docking that these mutations in real life should allow this compound to become a substrate. We should be able to change that selectivity. All right, there's a lot of data on this slide, so I'll just try to take you through a little bit slowly here. So this is looking at mutations in that enol reductase in vitro. What we've got down here are a slew of different possible substrates, okay? And each different colored bar corresponds to one of those mutant enol reductases that we've set up. And you can see, for example, the wild type is blue here. And so some of the substrates in the diketide is actually not a great substrate. The triketide is a much better substrate. But what you can really see is as soon as we start making those mutations, we're changing the selectivity of the enol reductase. And I just want to focus on one experiment in particular. If we look down here at this tetraketide, this doubly methylated tetraketide, it's not a substrate of the wild type. There's no blue bar here. We don't see turnover at all. In fact, it's an inhibitor. But in certain of these mutations, we can convert that now into a substrate. Okay, so that modeling and docking really seems like it pays off. It's a good model. It's predictive, and based on that model, we can make rational changes. So we can change the selectivity. Okay, so that's what we've done in the sort of divide and conquer area. I want to look now just at some of the other experiments we've done, which is building these chimeric systems. And we're going to focus on two different PKSs, which are going to be the kind of the ammunition, if you like, for these experiments. So here we're going to do our experiments in the tenolin PKS Nrps system. So here we've got one of these highly reducing PKS systems. It's a trans ER system, so there's a transacting ER, and the regular ER is broken. And downstream of the acyl carrier protein, there's a single module of a non-ribosomal peptide synthase. And so the polyketide synthase is building up, in the case of tenolin, this pentaketide. It's doubly methylated. You can see there's a reasonably sophisticated program here. And then downstream of that, the non-ribosomal peptide synthetase is selecting tyrosine. The condensation domain is going to form the peptide bond. There's a dipeptide cyclase here, which is then going to form a 5-membered ring and offload at the same time to make this tetramic acid. And then in the case of tenolin, there are 2 post-PKS tailoring steps, both P450s, one of which expands the ring to make this pyridone, and the second one hydroxylates the nitrogen. We're not going to worry too much about those at the moment. What we're interested in here is the program of the PKS. Okay, set against that, we're going to use a differently programmed system. This is a system involved in making desmethylbasianin, this compound. See, the final compound is similar to tenolin, the intermediate is similar to tenolin intermediate. And the difference here is that for desmethylbasianin, we have a hexaketide, so a longer chain, and just 1 methyl group. Tenolin has a pentaketide and 2 methyl groups. So we've got a small number of changes here. And again, just to emphasize the similarity with the vertebrate FAS, I've lined up the domain structure. Here on top is the vertebrate FAS. You can see the offloading mechanism here is the TE in that case. Here for the tenolin system, we've got the same domains in the same order, except we've got a functional methyltransferase, of course. And the offloading mechanism now we can think of as being the Nrps. This is offloading the polyketide from the system. And the types of experiment we're going to do here for the domain swaps, we're going to use recombination in yeast. This is very, very quick, very, very easy to do. Basically, we chop out a piece of our target synthase. We bring back some sequence patches, if you like, sequence, and we bring back the domain we want to insert. We transform these into yeast, and yeast pastes this back together seamlessly, very quickly, so we can make any swap we like with nucleotide resolution here. We then transform this into a fungal host in Express, and we analyze by LCMS. Okay, you can see in a typical experiment here, we might get a main compound and also minor compounds. All of these are purified and fully elucidated by NMR. So all the structures I show you are real structures. Okay. So here then, just to summarize some of the experiments we've done, we thought that perhaps because the Nrps is the offloading mechanism, that perhaps this could control the chain length in the same way that the TE does in FAS. Okay, but that's not the case. We can swap the Nrps's completely in these systems, and actually the polyketide that you get in each case is the polyketide which is programmed by the PKS. So all of the programming information is within the PKS, if you like. We can swap those in either direction, it doesn't make any difference. We wondered what would be the effect of this trans-acting ER. Could we swap those? It's a pretty easy experiment to do. And actually, we can swap the ERs. It doesn't make any difference to the program. The systems work perfectly well, so these are not involved either. And we wondered what would happen if we just omitted the transacting ER. We should get a polyene. Okay, and in fact, that's true. We do get a polyene. But in this experiment, the polyene turns out not to be the major product. We get a whole slew of products. It doesn't just make one thing. We get a whole library of products here. And what happens in this experiment is that the PKS loses its fidelity. It loses its ability to make a single compound, and it makes a whole mixture of different compounds. Okay, and you can see we've got different chain lengths. We've got shorter chain lengths. We've got different methylation patterns, and we've got different numbers of methylations. We've got 3 methyls down here. Okay, so the program is not kind of inherent to the PKS. It's mutable, it's changeable. Here are some more focused swaps. Okay, so now what we're doing is we're building up swaps from the KS terminus, and we're adding in one domain at a time. We can add the KS on its own or the KS and AT. It makes no difference. Both of these systems make 100% of this blue compound, which is down here. This is pre-tenolin, so they have not changed the program. Okay, as we add domains, we can add the DH. It doesn't change the program, no difference. Okay, but as we get into the methyltransferase, then there is a difference. Okay, and this kind of makes sense. Now we're getting about 75% of the green compound. Okay, and this is a new compound then. It's got the same chain length as tenolin, but it's only got 1 methyl group. So this makes sense. The methyltransferase domain has its own program somehow, which it can display, but it's not the whole story because we still get some dimethylation happening. We can keep going and include that broken ER. It makes no difference to the program. But as soon as we get down here to the KR, Okay, then suddenly something surprising happens. We get this red compound. And the red compound is one in which now we've got a hexaketide with just one methyl group. So the one methyl group makes sense because that's the program of the desmethylbasionin methyltransferase. But we didn't expect the KR to be able to control chain length. And this is a complete swap from one length to the other. If we extend again to include the ACP, then as expected, there's no change in the program. We can make more focused swaps in the middle, just the C-methyltransferase. This kind of reiterates what we saw before. We see a change in the methylation program. We can change fairly large chunks in the middle. Multi-domain piece here makes, again, mostly the hexaketide. Or we can change the KR on its own. And here we get a mixture. So again, we've kind of destroyed the program a little bit. But if you look at what we're seeing mostly orange and red compound. These are the hexaketides, okay, and the minority will be the pentaketides here. So the KR is really involved somehow in controlling chain length in these systems. More recently then, we've really focused in those experiments we're swapping the whole domain. So something like 400 or 500 amino acids in those swaps. Here what we're doing in this big table, we're looking inside the domain. So for example, with the C-methyltransferase here, we've divided up into 8 subfragments. And they're as small as maybe 30 residues. The KR, we're dividing that into 4 subfragments, 50, 60, 70 subfragments, residues in there. And we're making those swaps in exactly the same way. And what you can see is in some cases, in the methyltransferases, for example, we just get a whole slew of different compounds. It's really complicated to figure out what's going on here. We're really just breaking the program. These synthases are broken. But in the KR, we can actually figure out that smaller and smaller pieces, okay, so in fact, quarter-3 of the KR, here we can swap that, and we get some pretty high selectivity for hexaketides in this system. And here we're also doing another set of experiments. We're now bringing in an even longer system. This is militarinone. This is now a heptaketide with 3 methyl groups. And again, we can bring in that last quarter of the KR, and we can really focus in on making heptaketides selectively in this case. And you'll see down here an even tinier piece. This is just 12 residues, and it corresponds to a single helix. I'll talk about that as we go along. Okay, again, all of these compounds are isolated and fully characterized by NMR, so we know what's happening. Okay, so we can do the swaps, we can get compounds, we can reprogram the systems. We want to start to understand how that works. And again, we're going to use information from the vertebrate fats to build a model now of what we think the tenolin system might look like. Here in the vertebrate fats, the methyltransferase is just a stump, it's just a residue of the methyltransferase. And we use this to build on a methyltransferase model. We also use the sequences from the tenolin KR, for example, to build a KR model here. And for example, we can map onto that model all of the little pieces that we've swapped, and we can start to say, aha, when we swap this piece, it doesn't make a difference to the program, or it does make a difference, or it just breaks the system. Okay. We can start to dig into those questions. Something that we wanted to look at were correlations between the structure and the sequence changes. If we line up the sequences of the tenolin synthase and the desmethylbasionin synthase, we can pick out all of the change positions, and we can map those change positions onto this model structure. And it turns out that almost every single change is on the surface, on the periphery, nowhere near any active site. That kind of makes sense. If we delve deeper and we look into the active sites, it turns out that there are very, very few changes in the active sites themselves. And this is good for us because it's really focusing our attention on a single position. In the ketoreductase, for example, most of those changes are focused on a single helix, which is in the active site. But if we look at the methyltransferase, which is clearly involved in methylation programming, there are no changes in the active site. There are no changes which are adjacent to the substrate or the cofactor in the active site. In fact, there are very few changes in the methyltransferase core at all. It's almost identical, and yet it changes the program. Here's just a focus on that substrate binding helix in the KR. You can see the sequence differences here. And that, in fact, this is the 12 residue swap that we made And this is the smallest swap we've made so far, which changes the program. Okay, so we can really focus on tiny pieces of this synthase. All right, let me sum up a little bit about what we've seen then. This is just a model of the vertebrate fats. And we know that KS, AT, and ACP, we can swap these, they're unselected, they're not involved. Methyltransferase, of course, is involved in programming, it has its own program, but it's not an intrinsic property. It's not through mutations in the active site, it's some kind of extrinsic function, which we don't yet understand. The Nrps, again, we can swap that, it doesn't change the selectivity or the programming. The dehydratase doesn't seem to be involved in the programming, nor is the broken enol reductase. But the Kr controls the chain length. And you can see then that the Kr and the methyltransferase are clustered together in the structure. We also know that that transacting ER is somehow involved in the fidelity of the system. When it's absent, the system loses its ability to follow a single program. And so my guess is that that transacting ER docks with the whole system and holds the conformation quite rigid. I think the conformational dynamics here are going to be very important. And so when that ER is present, the program can be enacted. When it's gone, then the fidelity is lost. All right, so putting all of this information together, this is a mechanism by which we think the program may well be enacted in these iterative systems. And what we're going to think about is what happens at this ACP-bound stage. So particularly at this beta-dicarbonyl stage, where the substrate has just been extended by the KS, and now it's waiting on the ACP. And at this stage, this ACP-bound substrate could be the substrate for different catalytic domains. It could be methylated by the methyltransferase, it could be reduced, it could be offloaded, okay, or it could be extended again. And so these catalysts, if you like, are competing for this substrate. Whichever goes fastest is going to win, and it's going to enact its chemistry. So for example, if the methyltransferase is the fastest at that stage, it's going to methylate. Okay, so we get a methylated intermediate. Okay. It doesn't usually methylate again, so now there's another competition, and now if the Kr is the next fastest, it's going to reduce to give the alcohol. And then this compound is now committed to beta processing. Okay. We know that the dehydratase is unselective. It pretty well takes what it's given as long as it has the right stereochemistry. The DH is going to make this enol compound. And of course, if the enol reductase recognizes that, then that can be reduced. If the enol reductase doesn't recognize that, as we see in the squalestatin system, then the compound is blocked. It can't go forwards in the program. Okay? And it's just sitting, waiting on the ACP for some offloading process, then to release it from the system. Okay. But if the molecule goes forwards, if the intermediate goes forwards, at this stage, then the KS and AT, you can see, aha, now I've got a normal thiolester. That can be extended, we're back to a beta-carbonyl, and we're back into the system. And we've got, again, then a different series of competitions between these catalytic domains. And now, of course, the substrate is different, and so that competition is going to have different outcomes. So we think it's this internal competition which is happening at different points in the biosynthetic program, which is responsible for the overall program. And this is going to explain how the Kr is involved in chain length programming. Okay, so let me take you through what we think is happening in the tenolin and the desmethylbasianin and the militarinone system. So, of course, in the early stages, the diketide is built up as the first intermediate, and this is sitting on the ACP. And of course, this can be the substrate for the methyltransferase, for the Kr, or it could be the substrate for offloading. And in the case of tenolin and desmethylbasianin and militarinone, the methyltransferase always wins. It's always the fastest, so we see methylation. Okay, and once it's methylated, Kr can then come in, and the alcohol is then committed to beta processing, so we get this fully saturated intermediate here. In other systems that we know about, for example, cyclopyrazonic acid, it seems clear that the Nrps wins, is the fastest at this step, and it offloads just at this diketide stage. But here, for the systems we're looking at, methylation happens. Once this intermediate is built up, it's again committed to being extended by the Ks and At to make this beta carbonate. And again, we've got a point of competition. So here, the methyl transferase and the Kr and the Nrps can compete. In the case of tenolin and militarinone, the C-methyl transferase is fastest. Okay, it wins. In the case of desmethylbasianin, now the methyl transferase doesn't win, the Kr is faster. Okay, so it goes into this manifold here. Again, it's then committed to beta processing, chain extension to make a tetraketide. And again, we've got competition at this position. Okay, here we see the Kr normally wins for the tenolin and the desmethylbasianin, but for militarinone, there's a third methylation, so the methyl transferase must be fast. So we've got to a tetraketide. Again, this is then committed to chain extension. We'll get to the pentaketide. And here then is the point at which the chain length control kind of manifests itself. We've got to the pentaketide, and again, we've got competition. In the case of tenolin, the methyl transferase doesn't methylate again, and the Kr does not recognize pentaketides. So this is just waiting on the ACP. And what we think is that the Nrps is just slow. It just picks off whatever's waiting on the ACP and releases it. Okay, so here we've got a pentaketide. It gets released. And then again, this is then committed to the chemistry it's going to do, dipene cyclase and release eventually. But for the other two, the Kr was active. Okay, and so this reduction happens. Beta processing is then committed. Okay, and then chain extension must happen after that. And so that's why the Kr here is involved in controlling the chain length, because of this choice at this stage here. Again here, the methyl transferase and the Kr do not recognize hexaketides for the desmethylbasianin case. So the Nrps slowly picks off the hexaketide, cyclizes it and releases it. And in the militarinone case, okay, the Kr can still work. So this now gets pushed forwards into the heptaketide. And again, I guess at the end of this process, the militarinone Kr does not recognize heptaketides, and therefore the system gets released. So this is a model that we're now working with, the way we think these work, and that's going to, I hope, help us to engineer these systems further in a rational way as we go forward. So I should finish really by thanking all of the people. This has been a very, very long project in my group since I started my career a long time ago. It's been funded over the years in the UK, also here in Germany by DFG, so I'm very thankful to the funders. Okay. I kind of listed the papers in which we published all of this and the students who've worked on this over the years, but recently a lot of the work has been done by Oliver Peach and Sen Yin in my group, and Zhao Long, who's a CFC visitor, so I thank them very much. Also, my colleagues in the UK, in particular, Tom Simpson, with whom I worked very closely in the early days, and Colin Lazarus and Chris Willis and Andy Bailey in Bristol, who helped set up those recombination experiments. Also, very early in my career, GSK, Brian Rudd and Barry Wilkinson, who kind of pointed me in the right direction on these interesting projects. So I thank them very much, and I thank also everyone for your attention.
Thanks a lot, Marcel, for this talk, which I think we were even able to stream on YouTube.
Oh, well, so I'm a star.
Thanks for that as well. Talks over for discussion. So if there are questions from the audience, please ask them loud and clear. We also have questions by Zoom, which Martin will read essentially. Yes, please.
Thank you for that amazing wealth of information. It was really fascinating. Maybe it's obvious, but in which way does the polypeptide program itself in the conformation enters the active site and
it's
like when itself block some functions, so it's not recognized by the KR anymore. That's why it's not the process.
Yeah, we have a model of the KR. I would say it's a very unsophisticated model, and we can dock substrates into that model, but they're very unsatisfying results, if you like. With the ER, it's a pocket. It's like a sock, okay, and you can really see the chain go into that sock, and it locates itself and takes up a distinct conformation. The KR active site is much more open. and it's very difficult to dock substrates and get consistent results that I would like to write a paper about. All I can tell you is that when we swap that helix, which is in contact with the substrate, we can change the chain length selectivity, but what the actual molecular interactions are, I could not tell you.
Can I first say it's really hard to understand the questions back here if you speak over.
Okay, then we put it in repeat.
My question
Okay.
is, what role does information dynamics play? We have static structures based on does that move? Is that important for the reaction? Does that control the outcome? What do you know about that?
Yeah, so the question is, what role do structural dynamics play? I kind of tried to stress this as we went along. Our models are extremely unsophisticated. We don't take dynamics into account whatsoever. So they're terrible models. Okay, but what I can tell you is that the conformation, that S-cis, S-trans conformation I talked about, that is supported experimentally and in the models, but I couldn't tell you anything else about what's happening here. It's undoubtedly very sophisticated, and we have not done experiments in enough depth to be able to address that.
Okay, then I have a question of Adrian, who is in Zoom with us. So first of all, I'm just reading from the chat function, nice work, and it's about KS specificity. So how much substrate selectivity do you think KS have with respect to the chemistry of the alpha and beta carbon? So when double bonds are formed, it seems that KS must be selecting for the unsaturated over the beta hydroxy chain. When ER are present, do you think KS gatekeeps for the fully reduced intermediate?
We've worked with two different systems. I think in the squalene system, where we were working with the ER, it looks like the KS does not extend the tetraketide which is stuck, which is alpha beta unsaturated. We never see that being extended. But in the tenolin case, the KS will extend alpha beta unsaturated intermediates. And there we assume that the KS is pretty unselective. It will extend anything it's given.
I think Adrian will be happy with the answer.
Another question? No?
No more question.
You have a question?
I would like to ask about your docking tool, whatever you use for the retrograde fast. Say, I mean, could you just give us some whatever hints? Let's say, I mean, do you think that you could predict, let's say, how well?
I think you have to take each active site in isolation. Okay, they're all different from each other. We got very nice results with the dehydratase. Okay, we could dock substrates back in there, and we could show that compounds we know are turned over, they dock in productive conformations. Compounds we know do not turn over, do not dock in productive conformations. The same with the ER. But if we look at the KR, it's just chaos. You know, we can't do sensible docking because the active site is just too open. We don't know what's happening there. Yeah, I would definitely say the models we've built are very, very simplistic. They are not sophisticated in any way, but they work. For me, that's a good model.
Yes, question. I have a question about the swapping. There's a lot of swapping, and I'm interested in how you design this interface, because they end up.
Yeah, that's a really, really good question. I mean, we've been doing these swaps for a very long time. When we first started to do them, we tended to do them at what you would think are the traditional domain boundaries. Okay, and actually that more often than not doesn't work. What you want to do is pick a position in which the sequence similarity is already very high, because you're making the least damage, is my rationale, and those tend to work a lot better. In these systems, we're starting with PKSs which are already around about 80% identical. And that's a very forgiving system. If we drop down to 60%, things go wrong a lot more often. So we're lucky that we've already got systems which are highly similar and yet differently programmed.
there more questions from the audience or from Zoom? If that's not the case, then let me thank that we share with me the thanks for Russell and also for Eric for the first session.